Charlie, comencem? Sí? Perfecte. Hola, moltes gràcies per venir avui al Friday Talk. M'alegro que hàgiu vingut. Avui presentarem en Tarang. I will just read because he has quite a long CV, but he will present the genome evolution associated with fisheries and aquaculture relevant traits. So Tarang is a visitor. He has been in the institute since this week, so we had made plenty of times, even with the awful weather that we had. So you will have to come back. But so far the last day was sunny, so we will take some sun after this talk. So I will just read. So Dr. Tarang Mehta completed his PhD at the National University of Singapore, characterizing Hox genes clusters. Just a second, because with the glasses I don't see. Hox genes cluster in jawless vertebrates and timing vertebrates whole genome duplication events in the group of Vairapta Venkatesh and Sidney Brenner. After graduating, he completed 2 postdoc positions in the groups of Federica Di Palma and Wilfried Hartje at the Earlham Institute, Norwich, UK, focused on characterizing genes, regulatory networks associated with adaptive traits in fish. Tarang recently became a group leader in computational biology at the University of Liverpool in 2024, where his research broadly explores the genetic underpinnings of vertebrate adaptive traits and disease response, with an outlook towards preserving organismal health and biodiversity. Since much of his work utilized multi-omics data to characterize genetic diversity and identify markers of economically important traits, he also focuses on developing novel omics and algorithmic approaches for analyzing complex genomes. So thank you so much, Tarang, for giving us this talk. And so the floor is yours.
Thank you. Thank you very much for the introduction. Also, thank you very much for inviting me here for this talk. Absolutely delighted. Like Laia said, I am from the UK. It's probably quite clear because I'm the only person wearing a t-shirt when the sun is out. So I'm obviously making the most of it. So yeah, I will give quite a broad talk today. We have a lot of research going on within the group in kind of various areas, but I thought I would select topics which are a little bit more, let's say, relevant to some of the work potentially that you could have going on within the institute. Laia obviously introduced me, but I did my bachelor's in Nottingham in biomedical science, and then thought, well, I want to pursue a more kind of research active career. So then did a master's by research in molecular cellular biology. As Laia said, I then did my PhD in Singapore in comparative genomics, and then came back to the UK to do a postdoc and senior postdoc at the Earlham Institute, which is in Norwich. So that's in East Anglia, it's about 2 hours north of London. And then finally, this took me to a group leader position in computational biology at the University of Liverpool, where I direct various amounts of research, like I said, but the research program that I'll focus on today relates to one aspect of fisheries. So what is the long-term genomic impact of climate change on fish populations, and then how can we develop tools, more so genomic tools, to future-proof fishery sustainability. I have aspects of research which I still continue with from my postdocs related to aquaculture. So here we try to look at what the genomic impact of environmental or extreme changes in aquaculture relevant traits. And again, how can we develop genomic resources to create environmentally resilient strains? And then finally, I'd like to think a lot of this ties into aspects of genome evolution. So what can we learn about how genomes and traits evolve under aspects of environmental change, and then how is this then controlled by various facets of genome evolution? So for example, gene regulation, as well as structural rearrangements. And so for the first part of this talk, I'll focus on the fisheries component, and more specifically a PhD project in the group led by Victoria Oldcroft. So Victoria just started in October, so obviously this is very preliminary work, and this is a collaboration with CFAS, and so they are the governmental arm of fisheries management within the UK, and this is ultimately funded by NERC under UKRI. And more specifically, the title of this project is Adapting to Change, so integrating climate projections and genomic tools to future-proof UK fisheries management. As we're probably all quite well aware, climate change is altering ocean environments, increasing temperature as well as salinity changes, and this is really impacting commercial fish species distributions. As we can see in the kind of waters in and around the UK, sea surface temperatures are predicted to increase come 2060, and we're going to see quite drastic changes then in salinity come 2060. And so we're expecting these conditions, so temperature as well as salinity, to worsen in each scenario come 2060. And in the North Sea alone, actually a distinct warming trend has occurred over the past 30 years or so, with the area now identified as a hotspot of maritime climate change. So what impact then is this having on the species in and around the UK? Well, warm water species are becoming more abundant, as cold water species such as sole are in decline, and this is a trend obviously expected to continue given these range shifts in sea surface temperatures as well as salinity change. And what we're seeing then more specifically is certain flatfish species populations are shifting. So for example, European plaice is shifting kind of northeast, and there's been a 52% decline in landings since 2001, and Dover sole is shifting more southwesterly, and then also there are a change in landings, and within the UK especially, we're seeing quite drastic changes in North Sea cod, shifting more northeast, and these have been in decline since the 60s. And so obviously these shifts have great impacts more wider and beyond obviously just the species decline, but they affect fisheries because they increase travel distances, and they complicate catch predictions, as well as population numbers, and obviously this then has an impact on sustainable fisheries because a lot of those catching these fish populations, one, don't really know where to go, and two, might be depleting populations which, let's say, are within or in recovery. This project obviously has importance within these areas, but also wider importance. So we want to future-proof fisheries sustainability, but of course tools are needed to assess the long-term impact of climate change. And so based on this recent study, we could be then facing the possibility of 20% fewer marine animals by the end of the century. The problem that we have, though, at the moment is that recent distribution models treat species as just single units. And so this, therefore, then ignores a population's evolutionary adaptability to environmental changes. However, genomics can, of course, reveal adaptability within fish populations linked to, say, temperature and salinity. So we look for novel techniques like studying genetic evolution as well as phenotypic plasticity that can of course occur across generations, and of course multi-omics approaches can really then help with this type of analysis. And one of these approaches is genomic offset analysis, which can incorporate genetic adaptation units linked to climate distribution projections. And so essentially what this does is that it estimates the necessary genomic changes that are needed for a population to adapt to climate forecasts. And then basically this is done by this approach, which was reviewed quite lovely just a couple of years ago, like we show for kind of two dummy populations here. Genomic offset basically quantifies the mismatch that you have, or the offset between a population's current genetic adaptations and those required to adapt to future environments. And so this is done by basically modeling the relationship between genetic frequencies as well as environmental variables across species range, and then projecting how much genetic change is then needed to match future predicted conditions. And the best way that I like to put it is basically what you're doing is assessing the genetic toolkit that a species needs right now, or think of it like your wardrobe as an example. What wardrobe of clothes is basically perfect for the current environment, and how would your wardrobe need to change as climate then changes. And so basically for this, we first start by characterizing species population structure, and then integrating this with different climate scenario models. And so we started here with sole, for the reason, as I'd kind of mentioned earlier, but as their range was shifting. And the main aim here that we started was to firstly identify environmentally adaptive loci within UK sole populations. And so we collected samples in and around the UK English Channel. Don't know how visible that is. Yeah. Those in squares are our baseline samples. Those in circle are mixed samples, so they could be mixing between areas or ICEs, divisions as we call them. Red are those which are sampled just 1 year earlier compared to those in blue. We call SNPs in all of these individuals, so we started with roughly nearly 250 individuals. By the end of it, based on filtering, we kind of get down to something like more than something like 140 individuals, and then identify 7.2 million neutral SNPs. As you can see from the genome-wide PCA, looks like there is no signal of population structure here. However, some preliminary analysis that we've done, which I won't really show you today, shows no evidence, of course, of population structure at neutral loci. However, we do identify some differentiation at adaptive loci, and more kind of finer detail analysis then identifies that there could be subpopulations there. So one specifically in the ICEs division 7E, and then some in the Western divisions of 7E and 7F. And so our next steps are, like I said, this is all very preliminary. The PhD student just started in October, and they've obviously made fantastic progress in this really short period of time. But Vic's next steps are to annotate sites of genetic differentiation within the sole populations, and then move towards developing these genomic offset variables models, to predict population level adaptability. And more specifically, we want to really then focus on these two then subpopulations. And so for that, we need to integrate climate models. Next, we focused on place, again with a kind of similar aim here of firstly identifying environmentally associated adaptive loci. Again, samples were from quite a similar area in and around the English Channel, on a kind of similar sampling strategy. That's really difficult to see. But we ended up with roughly a little bit over 200 or so individuals, and what we first did was we did an analysis just of our samples, baseline samples, identifying 6.5 million neutral SNPs. And then what we did was we integrated published data with our data from Iceland and surrounding areas. And again, so from the genome-wide PCA, you can see it looks like there are 3 populations there. And preliminary analysis also, I'm not showing it here, identified neutral and adaptive differentiation. We find this within different populations, so within Iceland, as well as one in the North Sea, and some in our subpopulation data sets of population substructure in and around the Channel study region. What's really interesting here, and then this then integrates with our next steps, is that it looks like from preliminary analysis that chromosomal inversions could be driving some of the adaptive differentiation that we see between these populations. So next steps, it'll be really interesting to first annotate these sites of genetic differentiation, as well as the inversions that could be driving some of this population substructure, and then move on to genomic offset model analysis to predict population level adaptability within space, place, more specifically. And so you're probably wondering then, well, what are the kind of expected outcomes? Where would we move with this research? Well, fisheries managers will be provided via CFAS with firstly kind of crucial insights into some of the potential distribution shifts of these species. We plan to move to other species such as herring, sprat, as well as pollock, and whether there could be population declines in the face of changing ocean environments. So really have this enhanced understanding of fish population, adaptive capacities to climate change, also then provide models of improved accuracy, projecting future distributions, and almost then abundance of fish stocks, and then kind of finally end with this almost a decision support tool with infographics of models of what are the winners and losers, basically, of climate change impact on UK fish populations. So yeah, this will be a project for 3.5 years. So yeah, really excited for the outcomes of that. And so I'll move kind of swiftly on to then another part of my research focuses on aquaculture genomics and genomic evolution. Again here, just to remind you, I'm really interested on the genomic impact of environmental as well as extreme change on aquaculture relevant traits. And so here the main focus of my work has been on tilapia, which is of the genus Oreochromis. It's native to Africa as well as the Middle East, and it's farmed in well over 120 countries or territories. It accounts for roughly 9% of the 49 million tonnes of freshwater finfish which was produced globally in 2020, and it's the third most farmed species of those that are farmed globally. And the beauty, I think, of tilapia just as a whole is that it can reproduce in a wide range of aquaculture systems, and hence explains why it's the third most farmed species. Of course, I've gone over the situation where climate change is leading to extreme weather events, it's decreasing freshwater resources, and therefore there's a pressing issue to develop aquaculture systems, and more so than strains which are resilient to saline waters, as an example, as well as temperature changes. And a way forward, or almost our hypothesis here, is that it's important to study these genomic bases of adaptive traits, which could allow for genomic selective breeding into farmed strains. And so that's where I wanted to start, really, because it's estimated that by 2050, food demand will increase by upwards of 50%, with significant challenges such as increased scarcity and reduced quality of land as well as water resources. And so therefore there's obviously an increasing need for animal protein demand, with a growing importance then of aquaculture specifically for this. Why I'm explaining all of this? Well, we had collaborations with World Fish, who have developed a strain of tilapia called the genetically improved farm tilapia, or otherwise they call GIFT strain. The founding population comprises of wild tilapia, or wild Nile tilapia should I say, from various regions, so more specifically Egypt, Kenya, Ghana as well as Senegal. And they also take then domesticated species of Nile tilapia from regions in and around the Middle East as well as Southeast Asia. And basically, through their breeding strategy, they produced Nile tilapia which had increased growth as well as increased production time. And so basically the GIFT strain specifically grows upwards of 35% faster than non-GIFT tilapia, and as well it grows to an increased body weight upwards of nearly 90% compared to the base populations that were used for the breeding strategy. So obviously it's a well-revered tilapia strain then globally. The beauty of it then, as I mentioned earlier, is that it's suitable for both small-scale as well as commercial aquaculture. However, what was quite interesting was early on during the breeding strategy of GIFT, there was suggestions that there could have been introgression of genetic material from another tilapia species, so not Nile tilapia, but Mozambique tilapia, Oreochromis mozambicus. And so we were obviously interested in this because we wanted to see, well, if there was introgression of genetic loci from Mozambique tilapia, then one, was there introgression as a whole? What level was the introgression? And has it then contributed in some way to this increased growth as well as any level of resilience that GIFT strains show? So what we did here first was to generate genomic resources to identify genomic bases that could be associated with traits of interest in GIFT. We published this just a few years ago. So we generated a chromosome level genome assembly of GIFT to specifically highlight regions of introgression. Like I said, chromosome level, so we had short read, long read data, as well as a genetic map. So then we could map it to chromosomes. We had a really high level of contiguity, or completeness, should I say, then for that genome. And what was quite interesting, so of this roughly 1 gigabase genome, we found that 11 megabases of introgression were found from Oreochromis mozambicus, so from the Mozambique tilapia. When we then mapped what was in this 11 megabases of introgression from Mozambique tilapia to these Nile tilapia, we found that there were 283 genes within introgress region, and these could be associated with immunity and growth. So again, that was quite interesting to see then how this almost, in inverted commas, accidental breeding strategy where there was some introgression of Mozambique tilapia looks like it could have been beneficial for the immunity as well as growth of GIFT tilapia. So these resources are obviously very, very important for future genomic selection, breeding practices, as well as then considering whether introgression of desirable traits from other tilapia species is potentially then a really interesting future strategy then for creating novel strains. In the same way, World Fish have also developed another strain, which is called the genetically improved Abasa Nile tilapia, or as they call it, giant strain. And this was a program initiated in the early 2000s in Egypt And this was more driven towards suiting a Egyptian as well as aquaculture market and those conditions. And they went all the way down to the 8th and the 9th generations of giant, because they found that those grew upwards of nearly 30% faster and heavier than commercial strains, and they achieved really high fish yields, and they had nearly 16% lower feed conversion ratios as well. What was really good was the uptake for this. They knew there was a demand, so nearly 40% of total Egyptian fish farms received and now use the 9th generation of giant strain, and that was by 2017, so that it's probably much higher now. What's interesting though was, again, there were soundings that there could have been introgression along the breeding strategy of this. Again, this is Nile tilapia, but there could have been introgression from another Oreochromis species called Oreochromis aureus. So we go down a similar approach. So we generate giant genomic resources to look again at bases that could be associated with traits of interest in giant, and also we wanted to look at potentially this level of introgression. We again generate a high quality genome assembly based on short and long reads. We didn't have a genetic map this time for giant, but we still achieved quite good level of genome contiguity and completeness. I mean, when I say good, the Nile tilapia reference genome is less than the Contigen 50 that we have here for giant as well as gift. We annotated nearly 40,000 genes versus the near 38,000 in gift and the 33,000 in Nile tilapia reference genome. What was interesting was when we then mapped these to linkage groups, we found that 6 had regions of introgression from Oreochromis aureus. So again, there was an improved strain here which had introgression from another tilapia species. What was interesting that we were able to identify the X and Y linked haplotypes for sex determination, and they had genetic variation which followed the pattern as what was seen also in gift, which varied compared to Nile tilapia reference strain. So it looks like there's then a differential sex determination strategy then in these improved strains. So again, as an outlook, that's quite important for world fish. It's quite important for their breeding program, and then improving breeding practices as they continue for desirable traits. So how do we then move from developing these just genomes now? It seems like standard practice now to develop a genome. People have been doing it for a while, but then how can you then gain a deeper understanding of other aquaculture relevant traits? Well, like I said, we start by first generating those genomic resources when they're not available, or the contiguity of these or completeness of these genomes is not to sufficient standards. And we then move on to how can we then look at some of these resources being important for enabling the study of aquaculture relevant traits such as growth, immunity, as well as sex determination. And then how can we then take that even further, potentially to look at other aquaculture relevant traits associated with, let's say, environmental tolerance. One approach, and this is not new, this is not novel, it's been going on for a while, is to first think about how you could resequence populations, a little bit like we were doing in our kind of UK fisheries work, map and then characterize genetic variation, and then identify signatures of selection in genomic regions which could be associated with adaptive differences. These work from over nearly a decade ago, did whole genome resequencing of wild tilapia as well as commercial strains. They identified something like 1.3 million SNPs. What was interesting is the majority of these SNPs, as we kind of, I suppose, a little bit more well aware now, was the majority in non-coding regions. And only 16 non-synonymous SNPs were found in this study in 2015, of which 45% were intronic and 19% were intergenic SNPs, which are under selection, and they could be associated in and around genes which are associated with reproduction, growth, as well as immunity. And so I've just shown two studies here. There's studies obviously in other systems which suggest something quite similar, that selection on functional non-coding regions has played a prominent role during the domestication or breeding process of Nile tilapia or domesticated Nile tilapia species. Again, there was a huge gap then in genetic and genomic resources, or genomic resources, should I say, for tilapia species. So we wanted to develop then more resources to robustly identify non-coding selection. And so we, based on this rationale, we had this study where we generated genome-wide resequencing data across 575 individuals with deep phylogeny of tilapia species, 27 specifically tilapia species from across East Africa, to identify 69 million SNPs in the Nile tilapia genome. And so what we were able to do was we were able to assess phylogenomic patterns of recent as well as ancient hybridization between tilapia species. I won't go deep into that. That's another problem that they have for sure within Africa of hybridization of tilapia species through unsolicited stocking or replacing populations. And we published this not so long ago in 2024, and what we were able to find was contemporary levels of hybridization between native as well as introduced species. So these are species not native to the area, which were then introduced to the area, and then hybridization occurred, as well as ancient hybridization, which could contribute to more modern tilapia diversity that we're seeing. So I won't go deep into this paper, but this paper's very useful then for our kind of follow-up study. And so previously I utilized Nile tilapia as an outgroup in this study. And so Nile tilapia is a cichlid species. Cichlid species are a prime example of adaptive radiation. I developed this approach where I focus on just a few focal species from different lakes in and around East Africa. So Lake Victoria, Lake Tanganyika, and then Lake Malawi, and then Nile tilapia as an outgroup. We generate gene expression data so we could look at gene co-expression evolution. We then start to look at regulatory components to ask the question of where within non-coding regions do we have selection acting upon these sites that could be driving the expression pattern or co-expression pattern of genes across tissues, and then how can we study this in an integrated approach to look at, well, how our gene regulatory networks, so genes interacting as well as transcription factors with one another, how are they being rewired between species when we compare against the ancestral Nile tilapia to those which have evolved under adaptive radiations? Do we see patterns of similarity, or do you see stark levels of rewiring? What we ultimately show, and we published this just a few years ago, we started with this first paper where we show that discrete variation within transcription factor binding sites in gene promoter regions could disrupt regulatory edges that are associated with gene regulatory network rewiring of cichlid adaptive traits. So these are then genes which are associated with adaptive traits in cichlids. And then how can we leverage then more information if we start to look in and around downstream gene loci. So for example, in the 3' UTR, if we start to then predict microRNA binding sites. So we started to look at them in these 3-node motifs, whereby a microRNA could repress the expression pattern of a transcription factor as well as a target gene, but a transcription factor can activate its transcriptional levels of the target gene. So how often do these 3-node motifs occur, and how do they then vary between these 5 species and in different tissues? Again, we published this not so long ago, where we show that positive selection acting upon regulatory mutations, this time not just in transcription factor binding sites, but also in microRNA binding sites, could be associated with gene regulatory network rewiring. And this is more prominent or more enriched to be found, especially in genes associated with adaptive traits, for example, the visual systems, which are a trait under selection in cichlid species. So I don't want to talk about this work too much, but it builds the hypothesis for this study that we did, where basically no study that we knew at the time had evaluated the cis-regulatory impact of non-coding variation towards adaptive traits in a tilapia phylogeny. And so a technique useful for evaluating this is ATAC-seq. It can robustly identify genome-wide chromatin, as well as transcription factor occupancy using very few or even single cells. Briefly, what you do is you prepare the sample to expose regions of open chromatin, and then where sequencing adapters are used, they insert into active regions, amplified, and then they're sequenced. This includes identifying actively bound, then transcription factors through transcription factor footprinting. Basically, the logic here is in ATAC-seq. The enzyme that you use, the TM5 transposase, is enabled to cut chromatin, which is occupied by proteins such as transcription factors. So then basically when you map the reads, protein binding is then visible as a depletion of the signal. And that's what's defined then as a footprint. You can then map that to specific transcription factors based on their motif sequence. Again, despite the robustness of ATAC-seq, no such study at the time had been performed to date on tilapia tissues or cells. So given this, the aim of our study was to characterize open chromatin as well as gene expression landscape of Nile tilapia gill tissues to identify functional non-coding variation associated with environmental tolerance. And so here specifically, like I said, nobody had done this before. Not many people even at the time had done it much in zebrafish either, but we developed an optimized ATAC-seq then from Nile tilapia gill tissues, as well as associated RNA-seq, so extracting RNA as well to identify regions accounting for gene expression associated with gill adaptations. We sequenced to quite high depth for both, especially for ATAC-seq, so we could actually identify signals of transcription factor footprinting. We used a kind of classic approach here, just map it to the genome bow tie and then calling peaks using MAX2, and identified nearly 300,000 reproducible peaks in the gill tissues of 3 adult male Nile tilapias. We focus on the gills, probably as you're all aware, owing to salinity adaptations that are important for tolerating seawater compared to freshwater fish, such as the efficient diffusion of water and ions. And so, like I said, since we previously found that genetic variation disrupts regulatory network interactions for tilapia adaptive trait genes, we then wanted to test whether functional non-coding SNPs could be associated with the regulatory activity of genes involved with environmental tolerance in Nile tilapia. And so we work on the hypothesis here that tilapia species with similar environmental conditions would have a conserved genotype. So for example, if we looked in a gene which we know is associated with salinity tolerance, in tilapia species, and then we start to look in a gene promoter region, and more specifically then at a binding site of a transcription factor that we know is associated from our previous studies. Do we find evidence of just complete conservation there between species, so in a deep phylogeny? So like I said here, similar environmental conditions of these, say, two tilapia species, we would expect to have a conserved genotype at these non-coding regions, and we therefore then suggest that there is similar control of gene function, of gill function, sorry. And then under a different scenario, we could take two species with dissimilar environmental conditions, and do we then find either a conserved genotype at the same loci of the same salinity tolerance gene as an example, or do we find a non-conserved genotype? Is there a SNP which segregates based on environmental condition? And this therefore could suggest that there's different control of gene function from this transcription factor controlling the expression pattern of a salinity tolerance gene. So based on this logic, we first associated accessible peaks to the expression of unique genes in gills. We identify that most genes, so 1,650, and peaks, so nearly 10,500 peaks of chromatin activity could be categorized to this group, so this high accessibility. So these regions are highly accessible in the context of accessibility across the whole genome, and these genes are highly expressed in the context of all expressed genes in the genome. They were categorized into this group. We then start to integrate 69 million SNPs from our previous study to ask the question of, well, in these regions, if we take the promoter region, do any of these SNPs fall within transcription factor binding sites that could be controlling the expression pattern based on their activity in the genome and then the expression pattern of the gene that they are linked to? And then basically what we find is just over 1,000 or close to 1,200 high accessibility, high expression genes, which represents 4% of all Nile tilapia genes that have genetic variation at transcription factor binding sites, which are active within the gill. And I show kind of one example here in this almost quite complicated figure. And this is an example of regulatory variation, which is shown in the promoter of the prolactin receptor gene. So this is a gene involved or known to be involved in tilapia salinity tolerance. And basically what we find is that there is genetic variation in the promoter region of this gene. So there we have the peaks of activity in 3 samples. That's then the gene in the promoter region. These are all of the footprints that are found within that loci, and then these are all the SNPs. So we have SNPs there. And we find this genetic variation specifically in the footprint of this STAT1 transcription factor binding site. So STAT1 is a transcriptional activator that can be phosphorylated via osmotic stress. And we find that this differentially segregates from this TT genotype. So all the ones with red circles are the species that have the TT genotype that cause the transcription factor binding site. So that transcription factor binding site, therefore, we suggest is present in all of these species with the red circles, to a TG or a GG genotype that we predict to break the transcription factor binding site, so therefore not control the expression pattern of this gene in the highly adapted extremophile species Alcalapia grahami, which is a really interesting species because it lives in a really extreme environment compared to all of these other fish and just fish in general. It lives in 40 degree, upwards of 40 degree temperatures, really alkaline environments. And so what we therefore suggest here is that STAT1 could be a regulator of this osmotic stress response gene in these species, which are adapted to more of similar environments. However, the loss of STAT1 regulation of this osmotic response gene by substitution could potentially allow gill adaptations to really extreme conditions along these gradients of salinity, pH, as well as temperature, specifically in the alkaline tilapia. Hopefully I have a PhD position which is advertised at the moment, where we could start to look at this a little bit further if we get the right student. So hopefully I've shown how generation of novel integrative data revealed these candidate genes, so one associated with osmotic stress, one associated with permeability changes, associated with salinity acclimation. Again, we find lots of examples of this. I just showed two here, and how variation at regulatory loci could be associated with these aquaculture relevant traits, like salinity as well as osmotic stress acclimation. And of course you can read more about this in the paper that we published here. I don't want to go deep into this, but basically we followed this up in a kind of similar strategy where we looked on a kind of 5-cyclid phylogeny on a similar approach, but we interrogate more tissues here. Specifically aspects of the forebrain, the retina, liver and the testes, carrying out ATAC-seq as well as accompanied RNA-seq. This paper is currently under review and in bioarchive. And again, we find these similar examples of motif binding which has been broken by genetic variation when we compare them across the genomes, and we do more deep learning based predictions then on whether a substitution or genetic variation, should I say, can break binding than more than could occur by chance. And again, we kind of then look for this substitution to see whether it segregates along the phylogeny. So here we found this substitution between Niloticus and M. zebra, which is a Lake Malawi species. So again, we look at whether that SNP segregates along the same tilapia phylogeny of those 27 species. And then we also look in the Lake Malawi phylogeny of those species which are adapted to different habitats and environments. And we find this example of this visual photoreceptor gene where MAF, this transcription factor, could be directing the expression pattern of this visual photoreception gene, more specifically in this O. niloticus, the Nile tilapia clade with these other two species, so allowing it to potentially adapt to more freshwater environments. But the genetic variation that we see where it segregates in another Lake Malawi species is in this particular species, A. macroclethrum, which has a specialized ecological niche, and it has unique sensory and morphological adaptations to that environment. So potentially this non-control of MAF of ACTR1 could be allowing it to adapt with regards to visual photoreception in its specific environment. So I suppose to summarize for that component, I've shown you how we were able to take gene expression as well as the chromatin accessibility data to associate regulatory factors to gene expression, predict these regulatory networks and interactions, and show how we could use genetic variation data to associate regulatory variation to genes which are linked to environmental or adaptive traits, and how this is quite discrete and it could drive salinity acclimation as well as visual adaptation of one tilapia species and then maybe cichlid species. And so all of these papers, I suppose, to summarize, suggest that selection, as I was asking the question, and something that we really wanted to test, was suggestion on selection on functional non-coding regions has potentially played quite a prominent role in adaptive trait evolution during domestication, breeding, as well as speciation. What's next is we want to continue developing these systems and network biology and more machine learning approaches, looking at whether we can predict these levels of transcription factor binding site breaking or gain of binding sites, do more species and population level analysis of more whole tissues, and then go towards more focused single cell work to tease some of these factors apart. can we then take these to challenge and stress experiments to again test the logic in the same way, especially in tilapia species where we have different adaptations in different species? Can we then functionally validate this through genetic engineering, specifically in, let's say, a cell line? So test some of these interactions, test whether genetic variation or discrete genetic variation can disrupt the expression pattern of a gene, and then link these back to adaptive environmental as well as disease-resistant traits. And I kind of finally want to end on one of the main reasons why I'm here in this project where we're looking more specifically at fish genomics under extreme environments, where we want to characterize the epigenetic basis of fish embryo adaptations to background radiation as well as gravitational changes. This is a collaboration that I have with Laia and people, Maria more specifically in her group here at this institute, as well as Cyril, who's in France. And really the kind of question that we want to ask is, I know it sounds potentially far-fetched, but it's very, very interesting from multiple perspectives if we can farm fish in space. Obviously, this is gaining a lot of traction now, right? So the BBC picked it up, as well as The Guardian quite recently, where they interviewed Cyril about this logic. And I suppose you're asking the question, well, why? Well, it's to sustain human missions beyond low Earth orbit. Basically, the health of crews on long duration missions to the moon or Mars could be compromised solely on freeze-dried food. So producing fresh, nutrient-rich food in situ is a fundamental requirement. The challenge here is that their pre-packaged, these lyophilized food, are very poor on nutrient level, so they don't have much omega-3s or B vitamins, which are crucial for maintaining muscle mass. And of course, then fish are an ideal solution for this. And so the ultimate goal is to create this fully autonomous aquaculture system that is able to recycle waste. This would minimize resource dependency, especially on a lunar base. So the system would begin with water, which could be sourced from ice in craters at lunar poles. Fish waste could then be channeled to a second compartment to cultivate algae. The algae could then feed organisms such as zooplankton. Solid waste would be produced by shrimps and worms, and then the objective is to have this kind of zero waste system over a 4 to 5 month autonomous cycle. Of course, this is really relevant for activities on Earth, so it has vital applications here. So we could convert a system on Earth to one for, let's say, the moon. Of course, on Earth, this kind of recycled circular loop aquaculture system is important, not just for preserving the environment, but for economic durability. And also it's really relevant then to potentially feed quite isolated communities on Earth itself. A fundamental hurdle here, though, is asking the question of whether embryos can really survive this journey. So do they have the capacity to develop normally under these environmental stresses? And so we use here this dual species approach, focusing on zebrafish firstly, because it's a modeling organism, well annotated genes, rapid development, really good for pilot studies, and then kind of move on to European sea bass, which is a really vital aquaculture species in Europe and the kind of the target for this lunar hatch program. And there could be then real world potential of this concept. And so really what I want to do is then isolate the specific effects of microgravity using a clinostat. But to do that, we really want to eliminate these confounding variables, of which one is background radiation. And so the Canfranc facility here, just close to the border between Spain and France, provides this unique environment of cosmic silence. So we developed this study whereby we focus, like I said, on zebrafish and European sea bass species in this environment in Canfranc, where we submit the embryos then to microgravity using this clinostat system for 3 days, and then we start to then look at genetic triggers, so focusing specifically on epigenetics as well as transcriptome, and then also measuring phenotypes of the embryo, so how viable are they after spending 3 days of being hurtled around in a clinostat. So specifically survival, of course, behaviour and teratologies. And so we developed this genetic design to firstly to isolate these genetic triggers. So we do these experiments, one under cosmic silence. So in the Canfranc facility, the control is obviously no microgravity, the clinostat is with microgravity, but we also do a combined experiment then with that, where we do the same thing on the surface. So this is now with background radiation, same control, clinostat. We have roughly around 60 larvae per species, DNA and RNA extracted, sequenced, and then we analyze the data to look at overlap between conditions at species. For the RNA-seq, I don't want to bore you with the bioinformatics or anything, but we use a kind of standard approach. We trim, map, identify differentially expressed genes. And then essentially on our first level of analysis here, in zebrafish embryos, we actually find that there are very kind of few genes affected under microgravity. So in the cosmic silence scenario, we only identify 38 differentially expressed genes, representing only 0.2% of all expressed genes. When we do the same experiment but on the surface, so with background radiation or surface radiation, we find more, so 1,780 differentially expressed genes, which represents 7% of all expressed genes. And then when we plot some of the data and look at some of the genes which are, let's say, highly up or down regulated, we find some quite interesting examples associated with, say, DNA damage response, fin development, as well as myelin sheath formation. And I suppose the main conclusions that we want to take from this is basically based on differentially expressed genes. The response to simulated microgravity, especially under cosmic silence, is remarkably focused, and under cosmic silence, there are only very few genes which appear to be affected by microgravity, suggesting almost an inherent resilience of these embryos to these quite novel conditions. When we look at the overlap, and so when we're looking at the overlap, you have to consider then we start taking the radiation aspect out of it, and we're then purely just looking at the effect then of microgravity, and we only find 4 genes that could be implicated with the effect of microgravity on these embryos. Again, these are some are associated with scaffolding functions, as well as immunity and DNA binding activity, as well as cell division. We do a similar experiment in sea bass, but for the logistics and the schedule, we created a little bit more simpler design. We do the same control clinostat experiment under cosmic silence, but on the surface we only just take the control, so don't do a clinostat there. But again, even with this, we find only very few differentially expressed genes. So for example, under cosmic silence control versus clinostat, we're only finding 85 differentially expressed genes. Even if we compare that clinostat versus the background radiation, we're only talking about 200 genes. Even if you just take the control, so just looking at the effects of background radiation, we're only finding 77 differentially expressed genes. But I think the key thing I want you to take from this is when we compare it between the two species in this cosmic silence of microgravity versus no microgravity, we only find, like I said previously, 38 differentially expressed genes in zebrafish, representing only 0.2% of all expressed genes that we find in our study. And similarly, in sea bass, we have 0.4% of all expressed genes. So in both scenarios, both species, we have less than 0.5% expressed genes which are affected by microgravity. We then test in sea bass embryos whether of these 85 differentially expressed genes, what they could be over-enriched for. We find genes associated with response to external stimuli. It makes a lot of sense to us, as well as defense response. Those are of interest. If we look a little bit deeper into these 85 and those some of those overlap with the silence, with the microgravity under silence versus the control on the surface radiation. We find a few interesting genes which are particularly associated with innate immunity as well as inflammatory response. And again, focusing a little bit further, we have acute phase or immune response genes, chemical stress, as well as stress involvement genes. So genes that are being pulled out are those associated with stress response. These embryos are, of course, under stress. So this is an expectation, but it's still very few genes. What's interesting is if we compare this to a study that Cyril did just a few years ago, not to the same level, but they did 10 minutes of simulated hypergravity as well as then 39 hours of simulated microgravity versus the 3 days that we did. We start to pull out genes similar to what they pulled out from just qPCR studies. So they looked specifically at this complement component protein, which was significantly higher in microgravity, as well as interleukin-1 beta. And in our 85 differentially expressed genes, we find ones which appear to be associated with the same cascade. So C7 is associated with this complement cascade with C3, and BCL3 appears to directly repress interleukin-1 beta. So from 2 separate studies, we start to find coherence in some of the stress response genes that are coming out. I've been talking for quite a while, so I apologize. And I should practice my talk. And so I'll wrap up now. But what I want to conclude with this study is that microgravity is affecting just a few genes, which is perfect, really, for us, because we're not seeing huge, or it appears, stress response in both zebrafish and European sea bass embryos. Some of these genes that we're pulling out are related to one, immune and inflammatory response in European sea bass, and in zebrafish related to DNA binding, immunity and cell signaling. So these are potential then targets or candidate genes to target to see whether potentially if they could be boosted for increased resilience to these extreme conditions. And obviously we follow this up with some complementary validations of qPCR of certain genes that we selected, and then doing some more focused pathway enrichment analysis to see, well, which pathways are potentially perturbed under these environments. Gone through a lot today. We definitely do more work beyond this as well in different projects, but of course this would not be possible without lots of collaborators, including the fantastic ones that we have within this institute as well. And finally, to end, like I said, I've got a new group at the University of Liverpool. I'm more than happy to discuss opportunities for PhDs or supervising PhDs, as well as collaborations and research grants focused in a number of these different areas. So thank you very much for your time, and I apologise for keeping you from your lunch.
Okay, thank you so much. It has been fantastic and very exciting, a lot of work. So maybe we can have time for 1, 2 questions. Someone want to discuss something? Any question? Yes, sure.
Thank you very much for your talk. In the first part, I don't remember, I think it was the place, the example that you put with the place. I think that you found evidence of 3 different populations, right? Based on genetic signatures. So the question is, like, are there enough differences in temperature between these different locations that would then make this applicable to a scenario of climate change? Or these differences are probably due also to other factors, like, for example, difference in diet or predators or whatever?
No, sure, 100%. And it's a really good point, and it's something that we can't control if they have differences in diets, let's say, or anything like that. But there are indeed differences within temperature of where the species have been sampled. So less so in some of these aspects of the English Channel, more so in these aspects. But then when we take available data from Iceland and surrounding regions, then it's quite stark. And this is obviously something that we would then be testing with our offset analysis, where we introduce these climate variables into it.
How do you disentangle adaptation versus phenotypic plasticity?
Yeah.
Because this is a mechanism that would probably play an important role in the short term.
No, indeed. No, certainly. And one thing that we really want to do is when we start in these scenarios, try to identify whether chromosomal inversions are associated with some of this adaptive differentiation. We start to then link genetic factors into it. But yeah, disentangling adaptation to phenotypic plasticity is difficult if we don't go any further.
Hey, thanks for your talk. I was interested in more the technical aspect of using 27 different species to identify SNPs, and then based on one specific species where you did the ATAC-SIC. What was the technical approach? Because I assume that you used one reference genome for all of these, or what were the challenges?
No, it's a good question. I glossed over that. Nile-tilapia. We use Nile-tilapia as the reference. So we use Nile-tilapia to call the 69 million SNPs against the Nile-tilapia reference genome. We use Nile-tilapia specifically because it's the best genome that we have amongst all of these species. Actually, a lot of these don't have genomes. We're going to be publishing a paper hopefully this year where we've sequenced not 27 tilapia species, but upwards of 10 to 15 tilapia species. So we're adding resources there. But in each scenario, we use Nile-tilapia as a reference. And again, it makes sense completely because at the same time, we generate our ATAC-seq and RNA-seq data of gill tissues from Nile-tilapia as well.
And the strains you mentioned before, the giant and the gift, it seems that they have a significant amount of different genes, no? Like 23,000 compared to 40,000. Yeah. But this is within those 27 species, not where you used one reference genome, or is that?
I'm not sure.
So one species from those 27 contains, roughly speaking, 40,000 genes. Yeah. Where the reference genome that you used to align them to has 23. So I assume there's a stark difference. And I'm wondering, how do you deal with that uncertainty that there will be indels and structural variations, which probably affect the SNP comparison you're able to do on that?
No, of course, yeah. So in any scenario where we are identifying SNPs, we obviously go down a rigorous approach of filtering. We use filtering for LD then specifically, but we require a certain number of mapped reads as part of our filtering process to ensure that the SNP we're confident with the SNP as well, and also using minor allele frequency, so removing rare variants then as well. But of course, it's 100% an issue when you don't have nice complete genome. So the argument is sometimes you start to bias these approaches, because essentially then what you're doing is you're taking some really distant species and you're mapping it to a reference. So obviously the SNPs that are called within Nile-tilapia are going to be many more SNPs compared to if you take a distant species and map it to your Nile-tilapia reference. But until somebody generates loads of lovely genomes, then this is what we're stuck with. I assume this number would reduce as well the more we generate genomic resources.
Thanks.
Thank you so much again for just sharing your research. Amazing amount of work. Congratulations. And yeah. next time you're coming here in Barcelona, you can give us more improvement of these research that you have so many. Yeah.
I'll do a shorter talk. I promise.
Okay.